Okay, so for my project, I decided to test the survivorship of common water fleas, also known as Daphnia magna, when exposed to various light levels. So for those of you that don't know what Daphnia are, they're basically small planktonic crustaceans that inhabit many freshwater bodies around the world. These organisms are pretty small, but they do play a very substantial role in the health of the environment for two reasons. One, because like most organisms their size, they support the basis of the food chain, so they act as a food source for all larger organisms that are larger than themselves. Another thing that they do for the health of the ecosystem that is pretty good is basically just in their diet. Their primary food source is green algae, and green algae, when it's left basically untreated, it can grow to excessive amounts, which is called an algal bloom, and that is very, very detrimental to an ecosystem because it basically just sucks out all of the nutrients that other organisms would need, and ultimately it'll just kill them. So that is really bad for an environment. Um, Despite their size, Daphnia are pretty prone to environmental factors. One being the ions present in the water. They're very susceptible to changing water conditions. And another thing that what I decided to look at for my project was that they're very susceptible to different light exposures. So in my experiment, I wanted to test the survivorship of Daphnia in various light conditions. I wanted to compare two cultures of Daphnia, one that receives an equal amount of natural light and no light, and the other that receives absolutely no light at all. So I hypothesized that after the standing testing period of 5 days for each triplicate, the culture of Daphnia that received an equal amount of sunlight and darkness would have more individuals alive than in the culture that received no light at all. So this was a video that I took of my Daphnia that I received. I just thought it was a pretty cool video that I should incorporate into this poster because it allows you to see them pretty clearly as they are pretty small organisms. So for the materials that I needed, there were a lot of materials for this project that I did not have at home, and thanks to Colby Sawyer College, I was able to get these items shipped to me. This consisted of a live sample of Daphnia, which came in a small jar, which I will show later. Um, a small plastic suction probe and a bag of food pellets for them. So upon receiving the materials and the live specimens that I needed, I immediately filled up 2 mason jars, which I had at my home, and I filled them with natural spring water, Poland spring water. And I did this because tap water, as I said earlier in the presentation, contains a lot of ions that will ultimately kill the Daphnia, and I didn't want any other factors interfering with my experiment. So after both jars are filled, I used the suction probe to withdraw 20 live Daphnia from the original container that they were shipped in. So I put 10 in each jar, so 20 in total. I then placed a single food pellet into each jar and then moved the jars to their respective location. One jar was placed on the windowsill in my sunroom in order to receive an equal amount of natural sunlight during the day and then darkness at night. And the other jar was placed on the top shelf of my closet, and that closet did not have a light, and it was completely shut for the whole period that this experiment took place, so there was no interference taking place with the experiment. So as stated, these cultures remained in the same place for 5 days, completely untouched. At the end of the 5 days, I took out the jars from where they were, and I totaled the amount of Daphnia that were still alive and how many had died and wrote it into a spreadsheet. I then emptied each container, filled it with new water, 10 new specimens for each jar, and a fresh food pellet for each jar. And it's important to know that since this experiment was a triplicate, I did this 3 times, so twice after the initial time that I did it. So these were pictures that I took of my methods that I used to do this experiment and my setup. So this picture on the left right here, those were the 3 jars that I used. This one was just, I basically moved this was the original container that they shipped in on the right. So I just moved them to a larger jar to be easier to use the probe and transport them to their respective jar. And then these were the 2 that I actually used in the experiment. Obviously, I moved them to their actual location. This is just where I transfer the Daphnia into. This was a suction probe that I had that allowed me to transfer the Daphnia from here to one of the jars. And then this is the bag of their food pellets that came with the Daphnia, that I would give them 1 food pellet for every 5 days. So upon reviewing my results at the end of approximately 2 weeks, that's how long it took to do this whole experiment, I noticed that both cultures of Daphnia had some form of die-off of their specimens. On average, I noticed that the specimens that have been placed in the mason jar that receive equal amounts of sunlight and darkness, which is the one that was placed on my windowsill in my sunroom, were able to sustain themselves for longer periods of time and had more individuals alive than the specimens that were placed in complete darkness did. I also observed that the specimens that were in complete darkness, which were the ones in my closet that received no light at all, had a much higher rate of die-off in the span of 5 days than the other culture did. So, as I stated before, I compiled like all of the counts into one spreadsheet, and I made a 2 bar graphs that basically displayed the data that I found. So as you can see, like there's 3 different colors on this graph. Red indicates triplicate 1, 2, orange indicates triplicate 2, and yellow indicates triplicate 3. So, like as stated for the methods, I did the same methods for both cultures. And I noticed that as the experiment progressed, the Daphnia in the light receiving culture, so this bar graph, what this bar graph shows, began to slowly die off. The average count of live Daphnia specimens on the final day of the combined 3 triplicates was 19 out of 30. And so for both cultures, I did them 3 times, and 10 in each, so 10 times 3 is 30. So out of all of the experiments I did for the light receiving culture only 19 out of 30 survived, which equals out to 63.3%, which was a pretty high number. That's what I expected. On the other hand, the average count of dead specimens across the 3 combined triplicates was 11 out of 30, or 36.7%. So as with the light receiving culture, I did the exact same thing with the culture that received no light. Again, green indicates triplicate 1, blue is triplicate 2, and then purple is triplicate 3. So I did the same exact methods as I did with the other culture. Nothing changed. And as the experiment progressed, I noticed that the Daphnia in each experiment rapidly began to die off, more so in this culture, the one that received no light at all, than the other culture. The average count for live Daphnia specimens on the final day of the combined 3 triplicates was 10 out of 30. So again, this experiment for the no light receiving culture is done 3 times. 10 times 3 is 30. So only 10 across the whole 2-week period survived, and that equaled just about 33.3%. So it was a pretty low number. And the average count of dead specimens across the 3 combined triplicates was 20 out of 30, or about 66.7%. So there's a lot more dead specimens in this experiment. So at the end of the 2-week period that it took to fully complete this experiment, I concluded that the results that I observed agreed almost exactly with my hypothesis. My hypothesis was that after the standard testing period of 5 days for each triplicate, the culture of Daphnia that received an equal amount of sunlight and darkness would have more individuals alive than the culture that received no light at all. So this is exactly what I observed in my experiment. So my observations completely lined up with my hypothesis. So going back to the graphs, based on figure A, which was the bar graph that showed the light receiving culture. So based on that graph, it is evident that there were many more specimens that were alive compared to the other bar graph that showed the dark culture. Another thing to note that I found pretty important and pretty interesting was that for the dark receiving cultures, I noticed that they had a much higher rate of specimens dying off than the other culture did. So what this means is basically like for each experiment for both the light and the dark, I started with 10 individual Daphnias. So for the dark, I would put 10 in, check back a couple days later, and see that there were maybe only 4 or 5 still alive. But as for the culture that was placed on my windowsill that was receiving equal amounts of sunlight and darkness, I would put 10 in initially, come back a day or two later, and I would see that maybe only 2 or 3 died. So there were a lot more alive in the sunlight receiving culture. So naturally, the biggest factors that I had to overcome for this experiment, given that it was a lab that had to be done at home rather than what we were initially planning to do before campus closed, was that I had a limited number of specimens that I had to work with for the course of 2 weeks. When I was originally planning this experiment, I wanted to have at least 20 to 25 per jar rather than only 10, but I only had a small amount of Daphnia, and I had to make work with what I had. And basically, if someone wanted to do an extension off of my lab, I would say maybe start with, like as I said, having more specimens. So like minimum maybe of 20, even upwards of 50 to 60 would be a good range because it would give you a lot more data to support your hypothesis. It'd also be interesting to look at other light sources. So for mine, I did natural light and just complete darkness. But if someone wanted to do an extension, they could do what I did, like place one culture of Daphnia in like by a windowsill that received natural light, equal amounts of natural light and darkness, and the other one could be placed under maybe like a heat lamp or maybe just a light that's constantly 24/7 shining on them. That would just give some more interesting data to look at, kind of like a different variation of this experiment. Yeah. And this was just a picture that I found that shows where Daphnia like to reside and where they inhabit. And these are my citations, and that's pretty much it.